This is part 3 of 3 looking at chapter 6, where we're looking at energy, enzymes, and general overview of metabolism. So in the first part of this chapter, we talked about energy and the two different forms of energy and kind of how energy works. The second part of this chapter, we talked about enzymes and how enzymes work. So now we're going to kind of put those two different things together and look at an overview of metabolism. And like I said in the first part of this lecture, this chapter is kind of the building blocks to help us understand the next two chapters looking at our two big metabolic pathways, cellular respiration and photosynthesis. So metabolism is just all of the chemical reactions that occur within a cell. And these chemical reactions occur in things that are called metabolic pathways. And over here, this is a picture looking at cellular respiration, so how we take our food molecules. So you can see we have in the blue, there's amino acids which help build up proteins. We have glucose, which is a type of carbohydrate, and then we have the lipids, which are the fats and the oils. So cellular respiration, we're going to take the proteins, carbohydrates, fats, and oils. We're going to take them through this metabolic pathway and break it down to get out ATP or to get out energy. So we're going to be looking at cellular respiration in chapter 7 and photosynthesis in chapter 8. So metabolic pathway, so far we've been looking at really simple reactions where we start off with reactant or our initial substrate. It goes through a step and we get our products. But in metabolic pathways, we have multiple steps to get our final product. So in this example, we start with an initial substrate. This initial substrate goes through one step to get an intermediate molecule, and we're just going to call that intermediate number 1. In this really simple reaction, in this example, we're going to switch one of our OH groups on the bottom hand of our substrate, switch it out for a phosphate group onto our intermediate. So the phosphate is in the yellow part of the molecule. This step is going to take an enzyme in order to do this step, and we're going to call that enzyme number 1. We're then going to take intermediate number 1, take it through the second step using enzyme number 2 to get intermediate number 2. And here we switch out the top right-hand alcohol group, the OH, with another phosphate group. So far this metabolic pathway, we have 2 steps. We're going to have 2 different enzymes for those 2 different steps. And then this metabolic pathway is just going to have 3 steps to it, so it's a pretty short metabolic pathway. The third step, we have another enzyme, enzyme number 3, that's going to replace our third alcohol group, our third OH, with a third phosphate group. And this is going to be our final product. So the main thing about these metabolic pathways is that they're going to have multiple steps to them, and each of those steps is going to have its own specific enzyme that helps to speed up that specific step in the pathway. So these chemical reactions that are occurring in these metabolic pathways, our example again just had 3 steps in the pathway, but you can have pathways that are 10 steps or even more than that, and we'll see that when we get into the cellular respiration and the photosynthesis. Some of these metabolic pathways are catabolic pathways. This results in the breakdown of a molecule. So we have a large molecule that goes through this catabolic pathway. It's broken down to make smaller molecules. These catabolic pathways are going to be exergonic. They're going to release energy. So they're going to have that change in free energy that's going to be negative, which is exergonic. It's going to be spontaneous. Other metabolic pathways are anabolic pathways. So these are where we're going to synthesize or create different or larger molecules. And this takes energy, so they're going to be endergonic reactions. So these endergonic reactions, we're going to couple them with exergonic reactions like we talked about in the first part of this chapter. So we can say that our anabolic pathways, we're going to couple with a catabolic pathway so that the 2 pathways can together be coupled and we can end up with something that's spontaneous that's going to occur. So like I just mentioned, catabolic reactions or catabolic pathways, this is where we're going to start with the larger molecule. We're going to break it down. So we break down our reactant. These types of reactions are used for recycling. So cells really want to conserve energy. They want to conserve resources so that they're not wasting those energy or resources. In addition, catabolic pathways, we use them to obtain energy for our endergonic reactions. So remember, we want to extract energy from molecules, and then we're going to use that energy to run our endergonic reactions. This energy that we're going to extract out of molecules, we're going to store it in molecules called energy intermediates. And these are going to be things like our ATP that we've talked about a little bit. So remember, ATP, if we hydrolysize ATP, it lets off energy, so that's 7.3 kilocalories. Other energy intermediates, we're going to have things like NADH, we're going to have NADPH, and then there's another one that's not listed on here called FADH2. So we're going to see these energy intermediates show up a lot in the next two chapters. Remember, these are just molecules that store energy for a very short amount of time. Our anabolic reactions, these are reactions that we're going to use to synthesize things. So they're biosynthetic, bio means life, synthetic, we're creating something. So we're creating different molecules that we're going to use within the cell. These are used to make our large macromolecules. So if you remember, we talked about really large carbohydrates and proteins and lipids. So we're going to make those really large macromolecules, or we're going to use these reactions to make smaller molecules that we cannot get directly from food items. So anabolic reactions, we take little building blocks, we're going to add in our energy to create our larger molecules. So just a reminder, all these chemical reactions are going to occur in these metabolic pathways. We're either going to have our catabolic, we're going to break down molecules and get out energy, and then we're going to have anabolic, where we're going to create molecules and use up energy. Remember, these two things have to be coupled together with each other. In addition to these separate pathways, generally when we couple these two endergonic and exergonic reactions together, we're going to end up with something called a redox reaction. And this is how we're going to look at our photosynthesis and cellular respiration. We're going to look at it from the viewpoint of a redox reaction. So redox reactions, this is where we're going to move electrons or hydrogens from one molecule to another molecule. So we're going to remove electrons or hydrogens from one molecule and add those electrons, hydrogens to another molecule. Oxidation is when we remove the electrons, so that's where the ox in redox comes from. The reduction part of it is when you add on electrons or hydrogens, so that's the re part in the redox part. So we're going to have oxidation and reduction happening at the same time to get our redox. So an example of this, we're going to have molecule A that has an electron on it. We're going to also have molecule B. So these are our reactants we're starting with. Molecule A, what's going to happen is we're going to take off the electron from molecule A, so we're going to oxidize molecule A, or remove that electron. That electron is going to be placed onto molecule B. So if we go over to our product, so now we have A that has the electron missing. That electron is then added to molecule B. So we have B with the E negative on there. So A was oxidized. Molecule B, we say, has been reduced. So our electron has been added onto it. And I know this is kind of hard to understand because we're adding something to B, but we're saying that it's being reduced. Think about electrons, like we talked about in Chapter 2. Electrons have a negative charge to them. So we're actually, I think about it as reducing the charge. So we're adding an electron, but we're making the charge more negative or reducing that charge. So if that helps at all, great. If it didn't, sorry. Try to think of kind of a way to help make sense of these redox reactions. So in these different reactions that we've been talking about, we have our energy intermediates. And I know I mentioned them a little bit, but just to kind of talk about them a little bit more. So energy intermediates, when your electrons are removed by that oxidation process, the electrons, they don't just jump on to the other molecule. They don't directly cause the reduction right away. Generally, these electrons that are removed, they're put onto energy intermediates to create either ATP or NADH, NADPH, or here's our other one, FADH2, that I mentioned that was missing on the other slide. So these electrons, they get removed off a molecule, they get put onto an energy intermediate, and then they're used to reduce another molecule. So this is energy intermediates are just kind of this intermediate step between the oxidation and reduction part. So these energy intermediates, we can oxidize them to make ATP. We can take electrons off during different synthesis reactions like the one that we just looked at. So these energy intermediates hold on to electrons, and then we can use those electrons to do other things. So I mentioned that energy intermediates can be used to make ATP. So ATP, again, is an energy currency within a cell, so it's a very, very important molecule. So ATP is on, if you look on the right-hand side, we have our ATP. So remember, we have our adenine, our ribose sugar, and then our 3 phosphates attached. Remember, if we hydrolyze our ATP, if it goes through hydrolysis, we add water to it, it's going to cause one of those phosphates to break off and energy to be released. So we have that exergonic hydrolysis reaction on the bottom right here. So this ATP that we just broke apart to get out energy, the ATP, it can also go through a synthesis reaction, which is shown on the top, where we get energy from one of those energy intermediates, and we use it to stick that 3rd phosphate back on. So we start off with adenine, diphosphate, 2 phosphates, and a phosphate group that's kind of out there. We take energy to put that 3rd phosphate on to make ATP. So we covered quite a bit about the hydrolysis part, the bottom part of the cycle, but the synthesis part of ATP is just as important. So we need to be able to make our ATP in order to use it. So 2 ways to make ATP, and we're going to go through these quite a bit in the next 2 chapters, so it's kind of an introduction to these 2 different methods. So we have the first way, which is called substrate level phosphorylation. So substrate, whenever you see the word substrate, you can kind of think of an enzyme. So substrate level phosphorylation, this is going to involve an enzyme that will directly transfer the phosphate from one molecule onto our ADP to make ATP. So enzyme is used to transfer our phosphate from one molecule onto the ATP to create that. The other way is called chemiosmosis, and osmosis, remember, that is how water can move across a membrane. So osmosis, think about this as there's going to be some sort of membrane involved in this second way. So chemiosmosis, this is where energy is stored in an electrochemical gradient that's established across the membrane. And we're going to use that stored energy in this gradient to make ATP. And I'll show you how that works. But first, on the substrate level phosphorylation, we're going to have an enzyme that directly transfers a phosphate onto an ADP to make an ATP. So here we have a green enzyme. In our active site of the enzyme, we have ADP. It has 2 yellow phosphates. And then also on the active site, we have a molecule that has a 3rd phosphate on it. So this enzyme is going to help to reach the transition state, lower the activation energy. It's going to allow that 3rd phosphate to jump from this substrate molecule onto the ADP, and then we end up with ATP on the right-hand side. So substrate level, remember, enzyme is going to be directly involved in this process. The second way is called the chemiosmosis. So remember, osmosis is the movement of water. Think about having a membrane in here. I know we talked about this in a previous chapter. So this is where your energy from your gradient is going to be used to make ATP. So what's going to happen is you're going to have a gradient established. You're going to have a bunch of hydrogen ions on one side of a membrane. So in this example, we have a bunch of hydrogen ions on the top of our membrane, and then fewer hydrogens on the bottom of our membrane. So these are ions. They're not just going to diffuse through the membrane on their own. We have to have an active transport that's going to occur, or a facilitated transport that's going to occur. So these molecules are going to want to go from high concentration on the top to low concentration on the bottom. In order to do that, they have to go through a protein channel, and this protein channel is going to be called ATP synthase. So based on the name, you can kind of guess what it's going to do. So we're going to take these hydrogens, they're going to go from high to low through our ATP synthase. As they go through the ATP synthase, they're going to synthesize ATP. So that's our chemiosmosis way to create ATP. Okay, and then just very quickly, we have a few kind of miscellaneous things about these metabolic pathways. So here is our pathway that we looked at at the very beginning of this section of this chapter. So these pathways are run by these enzymes. And sometimes cells want to turn off pathways or slow the pathways down, kind of like how we can inhibit or stop enzymes from working that we looked at in the second part of this chapter. We can also inhibit or stop these pathways from going. So there's 3 ways where we can regulate these pathways. So there's regulation at the gene level or at the DNA level. So we can turn off or on genes that code for the enzymes. So if your gene is on, you're creating the enzymes, the pathway can go. If you turn the gene off, then our enzymes cannot be created, the pathway is turned off. We can regulate it at the cellular level. So this is when cells can talk to each other. So we have cell signaling pathways like hormones, so testosterone or estrogen, for example. I'm not going to go into a lot of detail of that. The third way is biochemical regulation. And I'm going to show you this, specifically something called feedback inhibition, or it's a negative feedback. This is where the product of your metabolic pathway, it's going to go back to an earlier step, and it's going to inhibit an early step or prevent an early step so that we can shut down the pathway and our final product doesn't overaccumulate. So this is an example of this negative feedback or feedback inhibition example. So we have our initial substrate. It goes into the active site of enzyme 1 right here. And then we create an intermediate, goes into enzyme 2, keeps on going until we get our green final product at the end. This final product, it's going to go back to our first enzyme in the pathway. So if you follow the red dotted arrow, it's going to enter into an allosteric site. So this is a form of non-competitive inhibition. And it's going to cause the active site on that first enzyme to change. And then your initial substrate can no longer enter into the active site, and it shuts the whole pathway down. Then the very final part of this chapter is looking at how cells can recycle. So most of the really large molecules in a cell, their only existence lasts for a really short amount of time. So living things, they have to be very efficient with energy and molecules and resources that they have. And I know I've mentioned this a couple of times throughout this chapter. And specifically, if we relate this back to when we looked at cell structure and function, there were things called lysosomes within a cell. These lysosomes, if you remember, they contain certain proteins called hydrolases. And these are enzymes that are going to break down our proteins, carbohydrates, nucleic acids, and lipids. So lysozymes, remember, are the recycling center within our cell. They're going to help recycle, they're going to help to get energy out of molecules that are no longer needed, and then we also get resources that can be reused by using these lysosomes. So they're going to digest substances taken up by endocytosis, so you can break down things that are brought into the cell. But the important part for recycling is the autophagy, or auto means cell, phagy means to eat. Autophagy is where the cell can recycle worn out organelles. So for example, mitochondria can wear out. So what can happen is we have a lysosome that's going to engulf a mitochondria, break it down, and then we can use all of those parts and energy to make more mitochondria or to make other types of things that the cell's going to need. So this chapter is all about energy, enzymes, and metabolism. So again, it's kind of giving us an introduction to different types of things that we're going to be using in our next 2 chapters.